Hello everyone, welcome to week number 5 in Bio 120. Last week was a super busy week. You had lots to do, so let's see what we've got going on this week. As a quick review, last week was about metabolism, and metabolism is all the chemical reactions that happen inside cells. And we included in that this week, respiration. So we had aerobic respiration, anaerobic respiration, and fermentation. These are ways that organisms break down food to make ATP molecules. And we also talked about photosynthesis, which some organisms on the planet do by taking sunlight energy and building up their own molecules of glucose. And you discussed the relationship between the two sets of chemical reactions. So this week is all about cell division. And we're going to talk about that and the life cycle of a cell called the cell cycle. Yeah, which you see a picture of over there. So skim through chapters 10, 14, and 15. You don't need to read them in detail. The videos are good this week. They give you a lot of information fairly quickly. So take a look at those. Read the Module 5 overview. This will also help you. And you have discussion board postings, of course, this week. We're going to talk about cancer this week. I'm a cancer survivor. I've had 2 different types. I've had a type of skin cancer, not melanoma, thank goodness, but I've also had breast cancer. So I'm very familiar with this. And so your question this week is cancer a problem with cell division, or is it a genetic disease, a disease of genes? So what are genes anyway? Genes are pieces of DNA that code for different traits in any organism. So you have long pieces of DNA, and there are little segments of it that are called genes. Not everything in the DNA is a gene. There's a lot of DNA that helps to regulate and turn on and off genes and just be part of the physical structure of the molecule. So genes are pieces of DNA that code for traits, and cell division is when the cell replicates itself. Or is cancer a problem with both? Now, I told you last week, when anybody asks you that, is it this one or that one or both? You know the answer is both. Otherwise, they wouldn't be asking you that. And how would lifestyle changes lower your cancer risk? So, before we move on any further, let's talk about the cell cycle, because this gives you an idea of how a cell works. So we start at the 12 o'clock position. You can see my image over there to the right. I have diagrammed out the cell cycle for you. So you start up there at the 12 o'clock noon, right? And then you move into something called the G1 phase. G1 is a time of general growth, where the size of the cell increases, the number of mitochondria increases, the number of enzymes increases. And I'll talk about G0 in just a second. But the first phase of the cell cycle is G1. So that's general growth, where the cell just gets bigger and increases the number of molecules and organelles in it, if your cell has organelles. So general growth. And then you have the S phase of the cell cycle. S phase is synthesis, and synthesis means to make. This is when new DNA is produced. So the cell's getting ready to divide. It's making a copy of the DNA that's there. So that's the S phase. Then you have the G2 phase. G2 phase is when all the cell machinery that's needed for cell division has to replicate and get put into place. So now the cell really is getting ready to divide. And then you have M and C, mitosis and cytokinesis. And this is where mitosis or meiosis, if you have a sex cell. But anyway, if you're talking about mitosis, this is where the nucleus divides. divides. And so the DNA molecules, the new ones, separate from each other and move to the daughter cells. And cytokinesis is when the inner portion of the cell divides, besides the nucleus. Okay, so that's the cell cycle. I told you I would come back to G0 because what we explained so far is how a cell divides. But that really is not the job of a cell, to divide. It's to move those cells into the future, yes. But cells have jobs, and that's where G0 comes in. So at some point in the G1 phase of the cell cycle, the cell will stop dividing, will stop moving forward in the cell cycle, and it will do its work. So if you're a heart muscle cell, the whole point of you is to contract, right? To push blood throughout the body. If you're a skin cell, it's to create a barrier between your organism and the outside world. If you're a cell of the small intestine, your job is to absorb nutrients and pass those along into the bloodstream. Now, I'm using human cells as examples, but they're good ones so that you can understand how this works. Cells have jobs. So that's what G0 is all about. It's a point in the G1 phase where the cell stops moving forward in the cell cycle and does the work that it's supposed to do. So this is very important. Cells have jobs. That's why they're cells, right? Okay. One problem with cancer cells is that they never stop in G0. They just keep flinging around that cell cycle, round and round and round and round and round, and dividing and dividing and dividing and dividing and dividing out of control. Well, what's that got to do with anything? Then the cancer cells eventually take up space, take up nutrients, and outcompete everything else in your body sometimes. So that gives you an idea of what cancer does. But also genes are involved because genes are involved in even running the cell cycle. But there are other genes, proto-oncogenes, tumor suppressor genes. You will read about those this week. And they are very important in the cancer process too. So take a look at those, and then you'll have a better understanding of how to answer that first question in your discussion board postings. You'll also answer the question about environmental carcinogens and how to remove them from the environment. So a carcinogen is anything that causes cancer. It might be a virus. The human papillomavirus, HPV, causes cancer. It's been known to cause cervical cancer, throat cancer, etc., etc. So it's a virus that causes cancer. It's a carcinogen. And then when you have something like benzene, you don't want to breathe in benzene from gasoline vapors because it causes cancer. Asbestos causes cancer. Formaldehyde causes cancer. These are carcinogens. The sunlight, ultraviolet light in sunlight, causes cancer. So there are lots of environmental carcinogens, and there are usually pretty good ways to get rid of these in the environment or protect yourself from them in the environment. So those are your discussion board postings. Then, let me move my face over. There you go. You have milestone 3 of your final project. You did milestone 1 in week 3, milestone 2 in week 4, milestone 3 in week 5. Okay, this is the third part, and this is really the last part of your final project before you put it all together. So here are the sections. Remember to use the outline template in the guidelines so that you're doing this correctly. You should have these headings included. Methods of conservation. So whatever your issue is, whether it's air pollution, eutrophication, climate change, whatever it is, you're going to cover this section, methods of conservation. And this means how can you prevent this issue from occurring? What are the things that we can do to prevent this situation? How can we prevent deforestation? We can stop cutting down trees. I mean, that's pretty simple. Yeah. But of course, we're going to have to find out why we're cutting down trees so we can stop doing it. And the next section is education. Why is education about this topic so important? Do you remember when you were in school some type of environmental issue that came up that you learned all about, and now you still do it? I mean, goodness, I've learned so many things over the years. You remember seeing commercials where sea turtles had the plastic rings from soda bottles wrapped around their necks or whatever, and everybody was supposed to cut up those plastic rings? Yeah, I still do that every time I have any of those. Yeah, so education is terribly important from school children on up. And then solutions. How do we fix the problems that are already here? That's different from methods. Methods is prevention. Solution is how to fix what has already happened. What are the ethical considerations? Are there any ethical considerations? Should people in poverty be more exposed to carcinogens, for example, than other members of the population just because they don't have as much money and don't have access to good medical care? You know, there are all kinds of ethical considerations. What are the laws in place? What are the regulations in place? What are the climate change treaties that are in place having to do with this particular topic? What are the positive and negative aspects of this issue? Are there any positives? Yeah, if you're talking about burning fossil fuels, it's just easier. We already have this whole system in place to burn fossil fuels. Aren't there gas stations everywhere? Yep. It's just easier. It's there. Now, with as much as it costs now, that's not so easy. But anyway, what are the negative aspects of this issue? And usually these are going to be numerous, because the whole reason you chose this topic was because it is a hot topic in biology today. And then finally, what can you do as an individual to help correct this problem, to fix this problem, to stop this problem? What can you personally do? What can everyone take away from this that they can personally do to help fix this? Include citations and references. This is very important. And this is the first time you will be making your topics here into slides, like PowerPoint slides. Now, you can make the slides in all different ways. There's Google Slides, PowerPoint, Canva, I don't know how else. I don't care how you make the slides. You can make them any way that you want. I most likely can open whatever it is that you send me. And if you can't, I'll let you know and we'll figure it out. So this would be 6 to 10 slides. Now look at how many topics you have. 1, 2, 3, 4, 5, 6. So you know you have to have 6 slides, one for each of these topics. Methods of conservation, right? Include graphics, include photos. Don't just type out a whole bunch of words on a slide. Nobody wants to see that. This will be the third part of your final project, so you'll already have this done, because for your final project you have to make a whole presentation of slides. So go ahead and add graphics. Go ahead and add photos, statistics, cartoons, drawings, you know, anything to make it interesting. And usually I recommend that you make a few bullet points. You can, there just went my light, oh well. You can put speaker's notes. You can put more verbiage on these slides if you have to, if you don't know how to do speaker's notes yet. I will add a video next week to show you how to put speaker's notes on slides. Or you can ask me and I'll tell you. So make PowerPoint slides, make slides this week. Label all your sections, like I already told you. Yeah, it's very, very important, because that makes sure that I know that you know what you're talking about. Come on. Oh, well. Well, that's not coming on. So this week is all about DNA. And if you want to skip this part, that's perfectly fine. This is a particular interest of mine, and it has been since the 1980s. Yeah, I've been doing this for a long time, and I've been interested in this for a long time. So, DNA fingerprinting. You've all heard of this. It's also called now DNA profiling, DNA typing. So what is this? This is trying to determine somebody's identity by looking at their DNA. That's what it is, basically. So this is Dr. Alec Jeffries. He's the one who came up with this particular technique, the first of these techniques. There's his picture down there in the bottom. And when he described to his wife what he was doing with his revolutionary study that he did, his wife said, that sounds a lot like fingerprinting. You know, when you're trying to identify somebody by using particular characteristics, why don't you call this DNA fingerprinting? And so he did, and that terminology caught on. It doesn't have anything to do with fingerprints. And now the name actually has changed because it's a bit different. So Alec Jeffries published an article in 1985 that described how he analyzed DNA, similar to the picture that I have over here on the right, to distinguish between members of a family and of a closed community also. So a lot of the people were very closely related, and he wanted to see if he could analyze DNA and figure out identities of people based on their DNA profiles that are shown over there. Those little dark bands, people say, you know, they look like a barcode, which they do back then. So he published this report in 1985, and some police in England read about his work and said, do you know what? I wonder if we could use this technique to help us catch a killer. And so they went to him. They said, what do you think about this? Do you think this could work? And he said, yeah, I think it could work. And they asked him if he would be willing to work with them. And he said, yes, because I would like to test this out. So the problem was that 3 young girls, 3 young teenagers had been killed in 3 different villages. They had been raped and killed. And the police thought this was the same perpetrator. The MO was exactly the same. And so what they started doing was going around and asking all the men in the 3 villages for blood samples. And in England, they don't have the same rights that we do. They can just insist that you give blood samples. And so that's what they did. And they collected over 5,000 blood samples. Now, I'm sure Alec Jeffries wasn't too crazy about this, because the technique was very laborious back then. Yeah, I learned how to do this, and yeah, very time consuming. And he started doing the technique, and he was comparing that to the DNA from the crime scenes. And he kept saying, nope, 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 nope. Haven't come across it yet. Nope, 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 nope. Well, if you're actually the person who killed these 3 girls, are you going to go in voluntarily and give your blood, even if you don't know what DNA fingerprinting is? No, you're not. And so the fellow, Colin Pitchfork, that is honest to goodness his name, convinced one of his buddies to go in twice and take his ID in and donate for him. His friend's name was Ian Kelly. And Ian Kelly, after this, was sitting around the pub and had a few too many and started bragging about what he'd done. And one of the women in the group said, you know, this is not passing the sniff test. I believe that I'm going to go tell the police about this. And she did. And the police tracked down and found Colin Pitchfork, a man who had a job, who had a family, who had children, never been in trouble with the law before. They tracked him down, took his blood, and sure enough, he had left behind the DNA at the crime scenes. First person convicted based on DNA fingerprinting, and it took off from there. In 1988, the FBI set up their program, and that was followed very closely by state police agencies in the United States setting up their DNA labs, and now pretty much everywhere has them, which is awesome. It's a much more streamlined technique now because we use a different technique, and actually it's become even more specialized than what I will discuss. So RFLP is what you see on the right, restriction fragment length polymorphism, and that just means the DNA is chopped up in different size fragments. That's all it means. So the barcode DNA fingerprint that you get over on the right over there is the RFLP technique. And how you read this, so what we have in this example is a mother, M, child, C, alleged father 1, alleged father 2. So the way to read this, you look at the child's DNA profile. So you start looking down the column, okay? You come to the first bar. And if there's a bar in the mother's profile, you just keep going. Then you look down to the second bar. Okay, there's no bar like this in the mother's profile, so it has to be in the father's profile. Do you see it in father 1? Nope. Do you see it in father 2? Yep. You keep going down. Keep going down in the child's profile. Yep. There's a bar over in the mother's profile. Keep going down to that thick one. Nope, not one in the mother's. Yes, there's one in F2. Okay, so what you figure out is old F2, alleged father 2, is the actual father to this child. That's how you read some of these barcodes. So along came PCR after this in the 1980s, the polymerase chain reaction. And this is just a way to multiply very tiny quantities of DNA that are out there. And so this revolutionized DNA fingerprinting even, because you needed a very, very small amount. It could be very degraded and you could still use it. And this is basically the technique that's used now because you're multiplying what is there. This was developed so now you can take more degraded samples, smaller samples, and take a look at the DNA. We're actually looking at single nucleotide polymorphism, so single differences in the base sequence of DNA between individuals to figure out somebody's DNA profile. That's what we use now, which is amazing that we can do that, but it has really come a long way since the 1980s. And then finally, in the last few years, this whole technique has taken a totally different shift through genealogy. So we have used sites like Ancestry.com, etc. You know, there are lots of these. And the information contained in the genealogy websites and the family histories and the family trees that are in these websites to catch criminals. The Golden State Killer, Joseph DeAngelo, was caught just a few years ago. He had killed people over 40 years ago. And he just aged out. He got too old and he couldn't physically murder people like he had. And so he quit. But he was still an awful old person. But he was caught by using forensic genealogy. And what is this? This is looking at the DNA through those genealogy websites. And what you're doing is narrowing down the person through looking at their DNA to families, really, is what you're doing. It's hardly ever true that you narrow it down to a specific person. You have to look at the family trees. You have to look at where these people have lived before. And you know, if you're talking about sexual assaults and rapes, it has to be a man. So you have to look for the sons in the family tree. You have to look at the age of them and when this happened, when the crime happened. So you have to use all these different factors, and then the police go and collect a sample from this person, either voluntarily or surreptitiously, like you can go through their trash and pull out a Kleenex from them and test the DNA from that. So this is the way that we're catching some criminals. Now, we're also determining unknown victims who are out there who they had some crime perpetrated against them, or they have just found a body in some location 50 years ago, and we are figuring out who they are doing this very same technique. So it's come in handy, and we have caught plenty of killers already doing this. So the DNA techniques have continued to move forward into the future. It's very exciting. And I've been interested in this for a long time. I did my PhD dissertation on part of this technique. And then I used it afterward in my research. So thought you might be interested in that. I'm interested in it. If you're not, just skip on past. I hope that you have a wonderful week this week. I know this will also be a busy week. Let me know if you have any questions, if you get stumped, like difference between methods and solutions, any of that. Just let me know. I'm glad to help. m.sigmon@snhu.edu. I'm here to help you. Have a great week.